let's just hop right in, and maybe you can share with me a bit about yourself and what motivated you to either be a part of this company or start this company.
I trained in PricewaterhouseCoopers as an accountant. So that's I suppose my qualification proper. Co-founded Inflection BioSciences back in 2012, and eventually, following a rigorous, comprehensive international global search for interesting opportunities, cancer opportunities, we reached out to 200 universities and research institutes and looked at 150 opportunities, and ultimately acquired the rights to the pipeline that we have today.
Maybe share a little bit, what problems does your company specifically address?
One of the most important pathways in cancer is the PI3 kinase pathway. Okay, so PI3 kinase is implicated in about 40% of all cancers. Okay, so it's a signaling pathway. So maybe I should ask, have you ever heard of the PI3 kinase pathway?
I have, but that doesn't mean our stakeholders have.
Okay. So it has attracted a lot of industry investment because of its involvement in so many cancers. The story with PI3K inhibition and targeting hasn't always been a promising one. It has been associated with side effect issues and the like. Nevertheless, it has turned a corner over the last few years, and we now have big pharma and others who have managed to get 4 different PI3 kinase drugs approved and on the market and treating patients. All right, so that's good news. That's great validation for the pathway. It's still not without challenge, and we believe as a company there's more to be had from targeting that pathway for the benefit of patients. And so this is where we come in. So what we know about PI3 kinase pathway is that resistance emerges, so the drug stops working. What we now know is that one of the key reasons that you get resistance is that there's an activating rescue pathway. So a parallel cancer signaling pathway gets activated as soon as you shut down. It's a bit like whack-a-mole. And this pathway is called PIM kinase. Okay, so we have shown, and Dana-Farber have shown, and MIT have shown, and University of Arizona have shown that when you shut down PI3K, you get a profound increase in PIM kinase, and it's like all of the messaging shifts over to PIM. And the papers and our work also would call then for a dual PIM and PI3K inhibition to get a more robust cancer kill and therefore more robust clinical outcomes and longer duration.
Let me ask a question. When you activate the PIM kinase, what occurs? What does that really mean?
So maybe if I'm in a room with private investors who have no knowledge of the space, I describe it like this. So maybe this is just a bit of a step back and might be too simplistic, but it's like I'm normally in a room and there's normally a big chandelier or a big light overhead, and I point to the light and I say, that's the cancer. And I say, we want to turn off that cancer. So you turn off the switch that is driving that light or that cancer. And I point to the switch on the wall and I say, that's PI3K switch stuck in the on position in the cells, giving signals for the light. So what normally would happen, you'll cut the wire, say, to that switch, and the light will go off, the cancer will go away, and you'll think, yes, we've solved it. But then all of a sudden, a few minutes later, it'll start flickering, and the light comes back on, the cancer's back, and you listen carefully, and you hear this murmur in the background, and that's the emergency generator kicking in, and that is the PIM kinase. So that's how I describe it.
That's perfect. That's perfect.
And there are other ways to describe it, and when I say it like that, our oncology experts always frown. But anyway, so PIM is really in our slide deck, we have a cartoon where it shows the 2 signaling pathways in the cells, and for somebody who's not used to seeing it, it just looks like a whole load of lines, and it's like a map, and what we show is that when you inhibit, there's a lot of bridges between the 2 pathways, and so when you shut down one, the cancer can still travel down through the bridges or the crosstalk, the shared substrates as they're called, I understand, and that takes over and can drive down through technically it's through the BCL2 pathway, and which is important for a number of different cancers, including B-cell malignancies. So it really takes over the ability for the cancer cells to replicate, which initially the PI3 kinase inhibitor would be stopping the replication of the cancer cells. And what we know now as well about that, this is a kind of a tangential point, but the PI3 kinase inhibitors generally don't kill the cancer cells, they stop them from replicating. So it's like you're storing up in a little jail all the cancer cells, and they're only waiting for a key to unlock that cell so they can get out and start. And that's what happens when something like the PIM kinase messaging comes along. It's able to get those cells that have been quiescent or quietened and can allow them to party again. So what we find with PIM kinase, because it is activating down the apoptosis pathway, it will actually intervene, and when co-administered with PI3K, it will actually kill those cells. So you get a significantly greater level of apoptosis. So that's a dual benefit of the PIM PI3K, which we believe should result in a more prolonged treatment effect, and which in our case will lead to longer patient survival.
Awesome. So who specifically is this targeted for? Who experiences this problem? And can you share maybe an example story of somebody who might experience this?
Okay, so I'll do my best. This is where perhaps our chief medical officer would have been a bit more articulate, but the PI3 kinase pathway is relevant in a whole host of different areas, as I mentioned earlier, but it is now clinically validated in B-cell malignancies.
And what are B-cell malignancies?
Okay, so B-cell malignancies are cancers caused by a cancer in the B-cell or a malignant B-cell where too many B-cells are allowed to prosper, and it smothers out the normal functioning of the cells, and you get horrendous side effects, and ultimately it's lethal. So
is B-cell a blood cancer? Is it a solid tumor?
Yeah, it's a lymphoma. Yes,
okay,
indeed.
perfect.
Yeah. So it affects about 700,000 people a year, and about 250,000 die every year. So although there have been wonderful advancements in treating B-cell malignancies or B-cell lymphomas, which are blood cancers effectively, people are still dying. So in particular, where do we come in? So right now, the PI3 kinase inhibitors are validated in B-cell malignancies. That means this is where they're approved, and this is where everybody agrees now they work. And they work through the B-cell pathway. So PI3K can interfere with the B-cell pathway to get a handle on the huge replication in B-cell cancers. The challenge with the PI3 kinase inhibitors is that they are used in the second line and third line because of their side effects. So normally somebody with a B-cell malignancy, traditionally they would go in for chemo or one of those standard treatment arrangements, and more recently then they'd go in for ibrutinib, which is a BTK inhibitor. So once these patients advance, because as all of these patients will get to a point where the first line will no longer work, and luckily the treatments are that good that one will get several years out of dealing with those first line therapies, be it chemo or ibrutinib. But when those options run out, you're left with PI3 kinase. PI3 kinase then will, on average, has been shown to extend life by somewhere between 9 and 12 months. And usually it stops. You only get that amount of time because of the resistance and the side effects. All right.
And what are the side effects?
Side effects range from severe pneumonia to hyperglycemia, to colitis, to severe diarrhea, so various GI issues. Many of those are treatable. As far as the clinicians will tell me that many of those are treatable, but they would only subject patients to that level of side effect if the efficacy justifies that level of and oftentimes the PI3K inhibitors, it gets to a point where that's no longer justifiable and the patients are taken off. And at that point then, oftentimes patients have no options left. So
Understood.
PI3Ks are kind of sitting in a situation where they're the last line of defense. So what can we do with our PI3 kinase inhibitors, or with our dual PIM PI3K? We can address the resistance that's caused by the emergence of the PIM kinase pathway. Okay, so we're the only company in the world that has developed a drug that will target at the same time the PI3K and the PIM kinase pathway. So in simple terms, many patients who would go on to a dual PIM PI3K should live longer than those who get a PI3K only inhibitor. We would also hope that the side effect profile of our drug would be superior. We would also hope that, given the data that we have, where we've shown that we're up to 50 times more potent than the PI3K only inhibitors, we would hope that that would allow a greater headroom between the efficacy and the safety, so that if the dose does need to be ramped up, if there is a wearing off effect after a year or more, that there is headroom to increase the dose. And then there are some other more specific examples and cancer settings where we've got very promising data, and I'll explain one which is in CLL, chronic lymphocytic leukemia, which again is a blood cancer. It's a leukemia, still within just about in the B-cell lymphoma space as far as I understand it. I don't know, 50, 60,000 people a year in the US get CLL. There are some wonderful medicines available for CLL, but one of the challenges is that CLL cells, they migrate into the bone marrow. Now, the bone marrow is a hypoxic tumor environment, microenvironment, so it's a low oxygen environment, and the studies have shown that the PI3 kinase inhibitors lose their effect in that environment. Okay. We have tested our drug head-to-head with the PI3 kinase inhibitors in a model of the bone marrow in this hypoxic environment, and we can show that our drug is still effective in that. Nice. So it could be that for those patients who are relapsing because of the pattern of the CLL cells homing towards the bone marrow, that our drug will still be effective. Furthermore, the PIM kinase itself has a particular function in the homing of cells into the bone marrow. So if you're inhibiting PIM, we have shown that you're confusing the CLL cells, or you're interfering with their ability to find that safe haven. So within CLL itself, there are a number of interesting settings where we should see a clinical differentiation versus the PI3Ks. So all told, this should benefit patients who maybe have gone through first line, and maybe the outcome is looking a bit more, relatively speaking, a bit more bleak at that stage. And this would be a brand new option which should provide more hope. So it's quite specific, it's quite targeted, and we would expect tens of thousands of patients would be candidates for a drug like this, and and possibly well more than that. I mean, because we have data across all the B-cell malignancies. I've mentioned chronic lymphocytic leukemia, but we would also see application in follicular lymphoma, which is another B-cell malignancy, also mantle cell lymphoma, also diffuse large B-cell and peripheral T-cell. And we also have interesting data and very strong rationale in acute in AML. That's myeloid, so that's not a lymphoma, it's a leukemia, right, where you have a 5-year survival, which is much, much worse than the lymphomas. So all that is on the table for our dual PIM PI3K. And as mentioned, we're the only company in the world that has a drug targeting both of these pathways. We will get orphan drug designation for this, and all going well, we will get accelerated approval to go into a single arm study. So it may only take as few as 100 patients in a final pivotal study to get initial approval for this drug in the States.
Nice. So is this an oral medication? Is it an infused medication?
No, this is oral. This will be a once-day oral drug.
And one of those that does it change things genetically so that if there are side effects, people have to live with those side effects, or does your research show that if they stop, those side effects go away?
Yeah, typically with the PI3Ks, that when you stop, the side effects will go away.
Nice. Okay.
So I think it's too early to call this, but a better way, as we look towards personalized medicine, and so that only those patients who are going to respond will get the drug, so that you're not exposing patients to side effects where they're not going to respond. One way we're looking at achieving that is in the initial clinical studies, we will measure the PIM kinase expression levels from each of the patients. And remembering that our drug is a dual PIM kinase, PI3 kinase inhibitor, it may well be that those patients who have higher PIM expression, when they're coming to the doctor and they already have high PIM expression, which goes hand in hand with B-cell malignancies. And indeed, as I mentioned, PIM expression is a barrier to the therapeutic effectiveness of the PI3Ks. So it could be that we may be able to stratify the patients based on their PIM expression. So that would mean, say, patients only with high or elevated PIM expression would get the drug. And if that is the case, it could be much more targeted and would mean that the therapeutic index or the side effect profile would be even better managed for those patients. But we can't make that call till we get into the clinic. But given that, 50 or 60 to 80% of B-cell malignant patients have high PIM expression, it could be that we don't need to stratify.
Right. So, and maybe you can't answer this, but is measuring a PIM cell expression in B-cell cancers, is that a routine test that happens for a patient with this kind of cancer?
It wouldn't be routine yet, but it is doable, but it's not, I can't say it's routine because there are no approved PIM kinase inhibitors.
Okay, and so it's not standard of care to have your PIM cell expression measured?
I would say not. Now, again, that would be a question for our CMO, but unlike, for example, PI3 kinase, where they routinely check whether you have a mutation of the PI3 kinase pathway, because there are no PIM inhibitors approved, it wouldn't be on the top list, I guess, of the normal. Having said that, given that there are PIM kinase inhibitors working through the clinic right now, so these aren't dual PIM inhibitors, but these are just PIM-only inhibitors. Right. All of those companies, Novartis and Incyte, they will be, I guess, doing the heavy lifting on the tricks and the steps to most optimally test for PIM expression.
It's
so
nice
again,
when somebody else. It's nice when somebody else can do some of the heavy lifting.
That's right, and we can benefit from all the work that the PI3 kinase companies have done, right? Because they've charted the path for us in terms of how we get approval, so we can follow and piggyback on what they have done for an expedited development. Except for the fact that we'll be hopefully bringing more to the table with our juice.
So, what are you seeing right now as the biggest challenges?
Fundraising.
Okay.
Yeah. So, I think as far as I'm concerned, the team is there, the technology is there, the development plan is there, and all that work has really come together in the last 6 months. I think we have our ducks in a row, as they say. We just need now the fuel to get on with it.
Thank you for watching the Lowdown on Cancer presented by the Cancer Fund. To learn more about what's coming in future episodes, visit us at www.cancerfund.com. While you're there, check out past episodes and be sure to click Join Us to become a member of the Cancer Fund community. As a member of the community, you'll receive early access to new interviews as well as other announcements. Make sure to connect to all of our social media channels so you don't miss a thing. In addition to getting early access to new episodes of the Lowdown on Cancer, Cancer Fund members play a critical role in driving cancer innovation forward so that one day we can more than hope. We can actually beat cancer. Thank you for joining us on this journey.